Hello and welcome to another exam mini video. My name is Fisher Stines, and today we're going to be talking about everyone's favorite houseplant on Stera Delicioso. So this is a jungle species that has amazing diversity in what it can do. And if we look here, we can see an individual that has a growth habit that is an erect, that has variegated leaves, and also has all these holes and is highly dissected. The other individual is having a climbing growth habit. The leaves are entire, meaning that they're smooth, and they're non-variegated. Now, if we go down the line here, what we want to do is a parental cross of these two individuals. And it's important to point out that both of them are pure breeding, so we're guaranteed to get a chromosome that is erect, variegated, and dissected, as well as a chromosome that is climbing, non-variegated, and entire. When we cross these together, we get one from the female, one from the male, and get this chromosome combination in our F1 generation. Now, we want to do some probability calculations, so we need to do a test cross to tease out both of these chromosome combinations in our F1 generation. So we have that listed here in this simplified chromosome map. So on one chromosome, we have erect, variegated, and dissected, and on another chromosome, we have climbing, non-variegated, and entire. Now, it's important to note here that there are some differences in these loci, and that's going to tie into how our probability calculations are done. So the growth habit loci and the variegation loci are 19.3 centimeters apart from one another. And on the other side here, the combination of variegated and dissected, that distance in centimorgans is 17.03. And our calculated coefficient of coincidence is 0.75. Now, if we look at our possibilities here, we see all sorts of combinations that add up to 8. And the way we get that is we're looking at erect, non-variegated, entire. That's a crossover event in region 1. And if we go to our second one here, we might look at climbing, non-variegated, entire. That is a no combination event. So we have climbing, non-variegated, and entire here. So that's what we're talking about. And we're going to do some probability calculations now. So to get a direct crossover, we're going to multiply region 1, region 2, and the coefficient of coincidence. Now in our example, in Morgan's, that's going to be 0.193, 0.1703, and 0.75. Now, because there are 2 chromosomes and 2 chances for that double combination event to occur, we have to multiply by 0.5 to get the gamete. And if we're moving to the same thing at the bottom here in SC1, we have to subtract that double crossover event from our total, and that's how we get that probability. And again, like the same in the previous example, we also have to multiply by 0.5. So here are the results of our experiment, essentially. So we have all of our different phenotypes and genotypes listed here, and they're paired. So the most common occurrence is going to be for a no crossover event, just like the parents. And the least common event is going to be for a double crossover. And that's the most combinations that we'll see. Here are my sources. Thank you very much.